Hello, my name is Hasan Sbaihat. I'm working at the Institute of Neuroscience and Medicine, Forschungszentrum Jülich, and RWTH Aachen University, under the supervision of Professor Dr. René Neuner, Forschungszentrum Jülich and RWTH Aachen University, and Dr. Abdul Nasser Asi from Arab American University. So today I'm going to give a brief introduction about the multimodal imaging, including the magnetic resonance imaging and the positron emission tomography, in addition to the electroencephalography. And then I would like to focus on the functional magnetic resonance imaging approach. So first of all, let us start with the human brain. So the human brain is a complex organ in the human body due to its huge responsibility for our thought, action, feeling, memory, and experience of the world. So this human brain, including a lot of brain networks, and each one it has its own specific task, and they are working efficiently and continuously sharing the information in a very short of time. So, but the question is how to investigate and how to study the relation and interaction between these brain networks, and it could be by using different modalities. Here we are offering three different brain modalities. The first one is the magnetic resonance imaging, which is the best choice for studying the spatial resolution. And the second one is the positron emission tomography, and it's considered as the perfect choice for studying the molecular and metabolic information. And the last one is the electroencephalography, which can study the temporal resolution of the human brain. But the question is, what if we combine these modality together? Then we can observe the behavior of the neuron in our brains from studying different features like spatial, temporal, and molecular. And fortunately, we have this multimodal imaging scanner in our institute. But as I mentioned earlier, in this talk, I would like to focus on the functional magnetic resonance imaging approach. And in this slide, I would like to figure out what is MRI and why we are using the MRI. The first thing, MRI is an imaging technique, uses the strong magnetic field in addition to the radio wave and the computer software to produce a detailed image of the organ and soft tissue contrast within the different organ of the body. And actually the MRI has a lot of advantages compared to the other brain imaging modality. So first of all, it is a non-ionizing technique, so this is pretty safe and this is good. And then it's non-invasive. There's no invasive technique in the MRI and could be repeated many times. And this is actually useful for studying the stability of networks and such things, because we can measure the subject or the patient more than one time, and this is completely safe. And then it combines a good spatial resolution with a reasonable temporal resolution. So our neurons in our brains don't have any storage of oxygen or neutrons, which mean whenever they are supposed to be activated, the blood flow should be increases to that region to bring neutrons and oxygen. So here, as we can see in the picture, the binding of the oxygen molecule to the hemoglobin molecule, which called oxyhemoglobin, and it has diamagnetic properties, while the free hemoglobin molecule, without the oxygen molecule, which called deoxyhemoglobin, and it has a paramagnetic property. So these two, deoxy and oxyhemoglobin, they has a different magnetic susceptibility, and that signal could be measured, which is the bold blood oxygen level dependent signal, by using echoplanar imaging sequence. So to conclude that, whenever we have high activity of the neurons, then the blood flow is supposed to be increased, and the oxyhemoglobin supposed to be increased as well, and we got more T2 star signal and then a high MRI signal. And as we can see at the right side of the slide, so here I'm just showing the concentration between the oxy and deoxyhemoglobin within the blood capillaries. So we can obviously observe that much less oxyhemoglobin under the resting state compared to the task condition. Here I'm showing, so in fMRI we have two main different design. The first one is the task fMRI, and the second one is resting state fMRI. So in the first one, so it includes three different methods. The first one is a block design. In that design, we ask the subject to do some specific task for a period of time, then waiting for some time, then repeat the task, and so on and so forth. In the second one, event related. So the subject supposed to do specific task whenever or as soon as they realize the stimulus, and the stimulus could be like visual or whatever, anything else. And the third one mixed, it's basically combination between these two designs. And in the task fMRI, change in bold signal refer to the experimental design, to our paradigm, and mapping brain functional based on the brain regions. So, all the region which should be activated supposed to be related to the task in somehow. And for the next one, resting state, actually it's very useful, and there is no task needed to be done by the subject, and the eyes could be open and could be closed, and using this paradigm, the spontaneous activity could be measured, and the connectivity, functional connectivity between different brain networks and different brain regions could be measured as well. Here I'm showing the visual audible paradigm. It's a common used task paradigm for the cognition and attention in the event-related potential study. And actually, as we can see in the right side of the slide, so there is two types of circle with different color. The first one is the blue one, audible target, and the second one is the yellow. So here we are, after both the subject in the scanner, we are presenting this stimuli, and we ask the subject to do some task whenever he realizes the target one, the blue one, and this task could be counting something or respond by their fingers or do something else. And it's worthy to mention that the target supposed to be more or less 5%, and the frequent more or less 95%. So here is how we built our model. So first of all, we have to define the stimulus, each single stimulus at a specific time. So for example, here we have a stimulus at 4, 10, and 20. And then we convolute this stimuli with the hemodynamic response function. Afterward, we got our model. Then here we expected all the voxels that related to that task supposed to be fit in somehow with this model. So here I'm showing two signals. So the green one is, let's say, our model, and the red one is some voxel. Then whenever existence of the significant correlation between these two signals mean that voxel related to the task paradigm. And then we do just threshold and clustering. Then we got some activation in different region of the brain. So here we have an audible paradigm dataset. It consists of 21 healthy volunteers. They have done 5 different sessions. The first one is resting state, and the final is resting state, both resting state, and 3 different tasks in between. So here is the pipeline of the analysis task paradigm. So we start with the pre-processing, including brain extraction, motion correction, temporal filtration, spatial smoothing, and slice time correction. Then the general linear model, 2 different types of events were created, the target and frequent, then convoluted with hemodynamic response function, then 5 different contrasts were created. And after, as soon as we got the activation region, we just co-registrate this activation region to the high resolution T1 images, then normalize to the standard space. And further, we did a group level analysis, and 3 different contrasts were created: respond more than count, and count more than passive, and finally respond more than passive. Here I'm showing the result, whereas the contrast respond more than count, which mean the brain region, the activated brain region, showing the region were activated at respond condition but not in count. But the question here, how to define this region? So we used Harvard-Oxford Cortical Atlas, then we got the region that activated. So for instance, in the left side, left hemisphere of the brain, we got some activation in the pre and post central gyrus, in the insular cortex, and in the right side, we got some activation in the insular cortex and the post central gyrus. So then we are done with the task paradigm, and here I'm going to show some result from the resting state. But the question is how to identify the brain networks. It could be done by using the independent component analysis, which uses different mathematical and statistical algorithm, which show the different brain region, which is spatially separated but functionally connected. And here we have the result of 3 different brain networks. The first one is the default mode network, consisting of the posterior cingulate cortex and the medial prefrontal cortex. And then we have the central executive network and the salience network. Then here I would like to speak a bit about the functional connectivity. So basically the functional connectivity is we can find it by examine the correlation between the signal between 2 different voxels. So the existence of significant correlation between them, which mean they are functionally connected. And it could be done by at voxel level or at region of interest level. Here we extracted the Bolt signal time series from different brain region, which come from different brain networks. Then the significant correlation between the first and the second one, which mean the BCC and the right lateral prefrontal cortex are functionally connected. So it could be done by applying a Pearson correlation coefficient, then Fisher R to Z transform to improve the normality of the dataset. But the question is still, why the functional connectivity is important? So actually, the alteration of the functional connectivity between different brain regions is associated with different disorders, including Alzheimer's disease, depression, schizophrenia, autism, and traumatic brain injuries. So here, studying the connectivity between different brain regions, comparing between health control and subject, could be used as a biomarker for early diagnosis of such disease, like neurological or psychiatric diseases. Moreover, the change interaction between brain networks associated with some, here we have a study which shows the change interaction between the brain networks has been shown in the patient with the major depression disorder and post-traumatic stress disorder, in addition to the schizophrenia. So here I would like to show other measure of functional connectivity, starting with the degree centrality. So actually the degree centrality reflects the global functional connectivity. In other words, it seems like we're asking the voxel who has more friends. So here we examine the correlation between given voxel and all other brain voxels, and then somehow the DC is supposed to be calculated. Then, for instance, here the voxel number 5, which has 0.77 degree of 77 value of DC, has more functional connectivity to the other brain networks compared to the voxel number 4, which has 0.66. So here there is some result which shows like the mean of DC in default mode network more than the DC mean of compared to the pre-task resistant state, which mean the default mode network was more connected to the other brain networks and region compared to the pre-task resistant state. And then we have the regional homogeneity. Here it seems like we're asking each voxel who has more friends living nearby. And here we have the amplitude of low frequency fluctuation, which indicate for the regional spontaneous activity. And it could be done by converting the time domain to the frequency domain, then calculating or computing the amplitude. So the current work, we have this dataset, the audible paradigm dataset. So actually we wanted to examine how the task paradigm could influence the post-task resistant state by using these different parameters. I mean the functional connectivity, including the global and local, DC and ReHo. In addition to that, examine the regional spontaneous activity and as well as the interaction between triple network module. And actually we got a good result and we are going to submit a manuscript by the end of next week. And I would like to thank the PF for their PhD funding and Palestinian German Science Bridge and Palest as well, and all the people who was contributing in this work. Thank you.